Yeah, hello. Can you please explain how capillaries work? What are their functions?
Okay, so the capillaries, they are on the edge of the tissues. So let me open the window. So I hope you see my screen now.
Yes, we see.
So we have the arteries. So these are big vessels that bring the oxygenated blood to tissues. Then they branch into smaller ones, and these smaller ones, they are called so artery and arteriole. So they are called arteriole, and uh, they also branch into the smaller and smaller vessels. And finally, they are left with the only one layer of the blood vessel wall, and this is the capillary. So capillary, and in capillary vessel wall, we will have only one cell, one layer of the cells, and these cells would be the endothelial cells. So why do we need them? Because we have the edge of the exchange with the tissues. Okay, so everything that we bring with blood, so it's oxygen, sorry, oxygen, nutrients, hormones, vitamins, etc. So we bring them with blood, and somehow we need to give these things to the tissues. So here we have the tissues. And of course, the tissues are composed of cells. And these cells need to obtain all these nutrients, etc., from the blood. So how do we do that? We diffuse so just imagine, so this is the vessel wall, and here we have tissue, which are the cells. And here we have the bloodstream with all these nutrients. So we diffuse them to our cells, okay? And to diffuse it efficiently, we need to get rid of all other barriers like muscle cells, etc. That's why our capillaries, they have only one layer of the cells in their wall, okay?
So arteries and veins only carry the blood, but
Yes,
capillaries like provide with all these nutrients and oxygen, right?
yes, you're absolutely right. So they are meant to uh, kind of deliver the stuff to the cells and take stuff like CO2, waste, and other stuff that is not needed by the cells. Okay, so they are kind of to make sure that the blood transports the stuff to the cells and from the cells. Okay, and
Okay, thank you.
arteries and veins, they are only for transportation of the blood.
Hello. Could you
Hello there.
please explain why thrombosis happens and can we cure it or is it fatal?
So we have the insufficiency of our blood vessels, and the insufficiency, so let's go in blood vessel insufficiency. So this means that they cannot perform their direct functions, and the direct function is to transport the blood. So let's say our veins, they cannot, so in our legs, so this is the leg vein, and it cannot transport the blood anymore, and it stays in the legs. So the blood is stuck. So why it might happen? Again, it's because of the pregnancy, because the pressure of the embryon on the big veins, it kind of prevents the blood flow from the legs. Or the person has some problems with their muscles. Let's say so first pregnancy, and second would be the low physical activity. So let's say they don't do these physical exercises, so their muscles, they are not strong, and they cannot kind of provide the veins with a pump function. So whenever we contract our muscles, so the muscle contraction is the pumping of our veins. Okay, so this is equal to muscle contraction. So in this case also, we will have the stuck blood in our veins. And usually whenever we have a lot of stuff that is not moving, so you have cells there, so it's red blood cells, white blood cells, and platelets. So they all kind of get older and older. So the red blood cells, they change their charge prematurely. So let's say they are only 40 days old, but they change the charge, and thus they start clumping. So let's say the red blood cells here. So now we are talking about this place. So the red blood cells, they get clumped, and also they kind of stitch or glue other cells too. So white blood cells plus platelets, they also get clumped here. And these clumps, they activate the system of clotting of the blood. And within this vein, we will obtain a huge clot. So it's a clump of red blood cells, white blood cells, and platelets. Plus, we will have some clotting factors. So these are the coagulation system factors. And they will be activated because the platelets got activated. So the platelets, they will release some transmitters that will activate the coagulation system. And in this case, we will have this clump, which is usually attached to the cell, to the blood wall. But at some point, let's say that the blood pressure, it's raised all of a sudden. So let's say a person started moving very quickly, and the blood flow kind of increases. And in this case, this clump of the or the thromb, it will suffer the pressure from the below. Okay, and sometimes it may lead to the detachment of these thromb, and it will float towards the heart. So as you understand, so we have legs, then so this is leg vessels. Then they go to our inferior vena cava, and then get to the heart. So here we have heart. So let's say one of these thrombs, it appeared somewhere in the legs and was detached, so it starts traveling. And when it travels, of course, in this leg, because of this huge vessel, which was wider than usual vessels are, so the diameter of this clump is kind of huge, okay? And let's say that it starts traveling across these, along these vessels, and get to the blood. And somehow it gets inside the blood and then enters the respiratory circle. And in the respiratory circle, it blocks the part of the usual vessel. So let's say this, just a second. So we have the pulmonary artery, and this thromb actually blocked it. And in this case, it is very dangerous because it might lead to the necrosis, not necrosis, but most probably the stop of functioning of the parts of the lungs, and it might lead to hypoxia, then local necrosis of the lung tissues, and in this case also to the inflammation, so pneumonias, etc. So this is very dangerous, but it might be even more dangerous if these thrombos, they get to the brain or to heart. So let's say that they block the coronary vessels. So some part of the heart will not receive oxygen, and because it constantly contracts, of course, it will accumulate a lot of waste and will not obtain the oxygen, and in this case, we will have the infarctus. And this means the death of the cells, and sometimes it might be fatal for the person, so they die because of the heart attack. So how do we prevent the thromboembolia? So whenever we have the detachment of the thromb, we speak about the thromboembolia. And this is the free floating of the thromb inside the circulatory system. So how could we prevent it? We can prevent it on early stages, sorry, with the physical activity. Then the compressing clothes. So you have special type of pants that you can wear, and they will compress the legs so that the diameter of the vessel does not go too far, and the bloodstream will stay the same so that the thrombosis doesn't happen. We can also give the anticoagulation factors to prevent this activation of the clotting, or we can give the I don't remember the exact name of the drugs, but it is against the activation of thrombocytes. So it's not the anti-thrombolysis, it just prevents the thrombosis itself, okay? And we can, I don't remember the drugs, so we can also give the thrombolytics, and these guys, they dissolve the formed thromb inside the vessel, okay? And they usually do it in a very fragile way so that the detachment doesn't happen. So we kind of peel the thromb. So let's say this is the thromb, and we peel it layer by layer. And in this case, we reduce the detachment probability. So this is how to fight the thrombosis, and I hope it was clear.
Thank you, it was clear. Thanks.
You're welcome. So we have the question from Ayan. So how does heart defects occur? And is it only about the aorta? No, actually we have a lot of defects. So just a second. So the heart, it's an organ which is composed of many tissues. So we might have the connective tissue as the outer shell of the heart. Then we have the muscle layer, and finally it is the inner part of the heart is lined with the epithelial tissue. And of course, whenever we create the heart during the embryogenesis, so we can have some defects of this formation. So the structure might kind of be incomplete. So usually we might have, so let's say we have this intraventricular septum. So this is the wall between two ventricles. And let's show it like this. We also have a thinner wall between two atria. So this is the right atrium. So this is the right ventricle. This is the left ventricle. And here we have the left atrium. So what are the problems might be with the heart structure? We can have incomplete formation of this septa. So this wall might have the hole in it, so that the blood from right side can go to the left side and vice versa. So this leads to hypoxia, for example. So people suffer from the low levels of oxygen. Why it happens? Because the oxygenated blood from the left atrium is mixed with the deoxygenated blood from the right atrium. So people can survive this condition or this defect, but they usually are very hypodynamic and apathic because they don't like the physical exercises, etc. So they feel dizzy, they cannot breathe as much as they want, and this leads to the feeling that they don't like the physical exercises. Next, we might have the problems with the wall between the two ventricles. The same goes, but usually these type of defects, they are much harsher, and people do not survive these type of the defects. We might also have the problem with the valves. So here we have the tricuspid valve, and here we have the mitral valve. So whenever we have problems with these ones, usually they are about the insufficiency of these valves, and they can give the way to the blood to get backwards. So it means in a usual way we have right atrium to right ventricle, and because of this insufficiency of valves, the blood gets back, okay? And this means that we have insufficient oxygenation of the blood. And here we have the same. So if we have the mitral defect, the deficiency of mitral valve, so not enough blood will get to the aorta because some part of it will get back to the left atrium. So in this case, we will also talk about the hypoxia in tissues, and you can clearly see and define these patients because they have kind of bluish skin. They are also apathic, etc. What are other defects? They might be also related to the aorta. So the aorta is the first vessel that is leaving the heart. So here we have the left ventricle, and here we will have the aortal valve. So the left ventricle is very, very powerful. So it can push the blood with a huge pressure and this blood hits the wall, okay? And here we have the very fragile place. And what are the defects of the aorta? We might have low elasticity, so we don't have enough elastic fibers there. Okay. And in this case, this part will suffer a lot of pressure, and at some point it might rupture because of the low elasticity in the wall. And the rupture leads to death because person just dies because of bleeding. We can also have the stenosis. So instead of usual diameter, we have the narrowing of the aorta, and this leads to lower bloodstream that can get through. And of course, it's also leads to the hypoxia in tissues, apathy, etc. What other? Okay, so we might have problems with the aortal valve, and again, it's about the insufficiency of transporting the oxygenated blood, etc. So as you can imagine, we, because of the complexity of heart itself and also the vessels that are coming to heart or leaving the heart, we can have a lot of problems, a lot of defects on the stage of embryogenesis. And some of these defects, we can treat them. Others are incurable. So they really require the how we say that, emergent or urgent surgeries, etc. So it really depends on the stage of the defect, on its severity, and on the patient's conditions. So if the patient can survive the defect, we usually don't touch them until they are about 18-year-old, because after 18, the whole organism of the person is developed enough so they can easily survive the surgery and intervention. But if the pathology is kind of harsh for the organism, we might take the person which is 2 days old. So it's just like 2 days after the delivery. So Ayan, I hope I answered your question.
I have a question about heart attack. You say that it because of the blocking of the blood, it occurs. But what about electric shock? What happens, like, to start beating when heart start beating because of electric shock?
You mean whenever it stopped and then we use this electric shock?
Yeah, exactly.
Okay, so it is called defibrillation, and usually it's not about starting the heart, which is stopped. We are trying to, so first, what is fibrillation? So we have heart, and it contracts in a way that you can define on the ECG. So we have the contraction of the atria, then we have the contraction of the ventricles, and then we have the relaxation. So you can define these stages on the ECG. So what we have in the fibrillation, we have spontaneous contractions of all parts of the heart in an asynchronous way. So instead of the usual spikes and peaks, we might have something like this, and we cannot define when we have the contraction of the atria, when we have the contraction of the ventricles, and when they are relaxing. So let's say at the same time we have the contraction of the left atria and right ventricle, and in a second or even less, we have the contraction of the right atria, and finally we get to the contraction of the left ventricle. So this is totally asynchronous. we don't have the synchronization of their work. So in this case, of course, the heart cannot perform its function. So it is the pumping of the blood, right? And we don't have pulse. So if you try to look at the person's pulse, you cannot sense anything because it's like this. And of course, with your hand sensing the pulse of the person, you will not feel anything, but you will see something on the ECG. And what we do, we usually give this electric shock, so the defibrillation. And it means that we kind of apply this electricity in the direction from atria to the apex of the heart. So through atria to ventricles to the apex. So this is how the electricity goes. And in this case, we will have kind of resetting of these contractions. So how we do that, we just give this controlled depolarization of all cells in the heart, and we give it with this electricity. So at this point, our heart stops moving or stops contracting. And this leads to the restart, or if you think about the computer science, it's rebooting of the conduction system. So here we have this pacemaker. It kind of reboots, restarts, sorry, restarts and starts sending the rhythmic signals to the heart. And then we might have the usual picture or usual image on ECG. So if we got the usual picture, we stop with the electricity, etc. And we say that, okay, so the fibrillation stopped, and we can continue with the usual treatment, etc. But if we still see the fibrillation, we increase the electricity, or the power of electricity, and give the same shock again, and look at the ECG. So we monitor the picture of the ECG.
Please, can you explain about coronary circulation and also about systolic and diastolic pressure?
So, okay. So first, it's about the coronary circulation, right? So we have
Yes.
the aorta, and so here is the heart, and from the aorta, right at the beginning here and here, we have two small holes, and these holes open into the coronary arteries. So we have left and right artery, and let's say that, so here we have this valve. So this is aortal valve. And let's say that the blood is pumped in this direction. So when the left ventricle is contracted, the blood gets to the bulb of the aorta. So this part. So this part is called bulb. And the blood stays here. Okay, so after the valve is closed, it cannot go in this direction. So usually it goes into this direction because we have the barrier of the valves. And the aorta kind of gives the way to the blood to go to smaller arteries. But the blood actually tries to get back to the ventricle, and because it's stopped here, it also gets to the coronary arteries, And in this way, we have kind of high blood pressure inside these coronary arteries. And this blood pressure gives the possibility for our heart to obtain a lot of nutrients, a lot of oxygen, etc., in a very short time. So it is pushed to all the branches of the arteries. So it gives the nutrients and oxygen. Then it is collected by the venules, then get to the venous system of the heart, venous sinus, and is opened directly into the vena cava. So we have vena cava superior here and vena cava inferior here. So inferior means the one that is below, superior means the one that is above. So we have the venous system of the blood, of the heart, that kind of flashes the deoxygenated blood to the vena cava. And it gets to the right atrium, then to the right ventricle, and then into lungs for oxygenation. So this is the coronary cycle. And is it clear now? I'm sorry, I don't remember who was asking the question.
Yes, it's clear. And can you
So it's
explain
only about the coronary.
Mhm. And can you please explain about systolic and diastolic
Yes, of course,
blood
I'm getting
pressure?
to it. Yeah, so just a second. So we have the, so it's still bulb, and you have the blood that comes from the left ventricle. So left ventricle pumps the blood here, okay? And at the moment of this pumping, so the pressure inside this bulb of the aorta is systolic. Systolic. I don't remember how it's written. So yes, so we have the systolic blood pressure. So it's the maximum that we have. So usually we have 120 or so. So this means, so the systolic one is when the heart is pumped from the ventricle, okay? And when it hits the wall of the aorta, we can fix or we can write down the pressure that is inside aorta at this moment. So the diastolic, uh, after we pump the blood, we start relaxing. So the left ventricle gets into the phase of relaxation. And this moment, the left ventricle is kind of empty, right? So because it's empty and the pressure here, so let's call it pressure 1, and here we have the pressure 2. So pressure 2 is much higher than the pressure 1. And because of that, the blood from the bulb tries to get back to the ventricle because it's lower pressure there. And we go through the path of lower resistance. So we are trying to get back, but the aortal valve does not let us do that. So we have a very stiff and very rigid valve here. So it does not let the blood get back to this place. And most of the blood actually already gone through this aortal arc. And the pressure that is left here at the moment of the relaxation of the left ventricle is called diastolic. Okay, is it clear?
Yes, it's much clearer now. Thank you.
Nice, you're welcome. So this is the minimum, and let's make it 80 millimeters of the heterargon.
May I ask a question from the previous lecture?
Yeah, for sure.
Could you please briefly explain how muscle contractions occur?
So we have the filaments, and let's start with the actin filaments. So it is in the helix conformation, and we have some binding sites for the myosin. So this binding site means the place that the myosin can bind to. Okay, so it's quite simple. But we permanently have these binding sites occupied by the tropomyosin. So these tropomyosin here, so actin, tropomyosin. So it kind of blocks or prevents the myosin from interacting with the actin. But at the same time, it is already bound to troponin. And let's picture it like this. So we have troponin that is bound to the tropomyosin, okay? So what happens next? We have the, like this, the fibers of myosin that has so-called heads. And these heads, they are actually interacting with the actin. So it can bind to this binding site of the actin and just stay kind of glued to it, okay? So what happens? We have the nerve impulse, and this nerve impulse gets to the muscle membrane. So it's a cell membrane, and it releases some kind of transmitters or the signals that will let the muscle know that the contraction is needed, okay? So we have special receptors on the muscle membrane, and whenever these receptors get these transmitters, and in this case it's acetylcholine, so they bind to these receptors and send the signal inside the cell, and this signal is responsible for the okay, so for the release of calcium ions that are stored inside the sarcoplasmic reticulum. So we have the sarcoplasmic reticulum and calcium ions inside it. So this signal affects the sarcoplasmic reticulum and it releases the calcium ions. So these calcium ions, whenever their concentration is increased inside the cell, so in cytoplasm, they start binding to our troponin. So here we have the, let's call it like this. Whenever we bind the troponin to the calcium ions, the tropomyosin troponin complex, it changes the conformation. So conformation change. And in this way, the binding sites, they get free. So they are not blocked anymore by the tropomyosin complex. And since they are free, the myosin fiber, it has the access and it really starts moving towards these binding sites. So let's say that at this point, we are bound to this binding site. So the next step would be taking the ATP molecule and binding it to the head of the myosin. So the ATP is bound now, and the myosin, it actually breaks the first bond of the ATP, so it hydrolyzes it to ADP and one phosphate group. So after it hydrolyzes it, the both molecules, they stay on the head. Okay, so now we have the ADP and small phosphate. And in this case, the whole head starts moving. So instead of this position, it will look like this. So the first position without ATP was like this. Now it kind of unwinds. And in this case, it will kind of prolong itself towards the next binding site. Okay, so after that, we released one of the phosphates. And in this case, whenever we have only ADP attached to our head, and it is bound to the next binding site now. So it kind of moved from one binding site to another and released this phosphate here. After that, it releases the ADP2 and starts kind of contracting. So here we have the ADP and phosphate, and now it will pull itself to get the previous state. But in the case of the release of this ADP and phosphate, it kind of changed the position in space, right? So it was next to one point, next one side, and now it's bound to another one. So this means that we have the movement of two fibers across along each other. So we have, let's say, we have this actin and the myosin. That myosin moves kind of along the fiber of actins. Okay, so that's how the process of contraction happens. So it's only related to the movement of myosin along the actin. That's really simple. So I hope you understood it.
Yeah,
so was it clear?
It's a bit clearer now. Yes, sure, thank you.
Okay, nice. You're welcome. So of course, whenever you don't have the acetylcholine release anymore, so this signal stops, and the calcium from here gets back to the sarcoplasmic reticulum, and the opposite action happens. So the myosin slides back again along this fiber of actin. And in this case, we speak about the relaxation of the muscle fiber. So it's just sliding of myosin along the actin. So it's just the opposite process of the contraction. Or we might also, instead of acetylcholine blockage, we can also run out of our ATP. So if we don't have enough ATP, again, the sliding of the myosin along the actin happens. So if we don't have ATP, again, the muscle fiber is relaxed again. So this is how it works.